Everyone, and welcome to today's webinar, Beyond the Surface of Neuroscience, 3D Imaging of Large Biological Samples with Light Sheet Microscopy. I am Cassie Soltman of Labroots, and I'll be your moderator for today's event. Today's educational web seminar is presented by Labroots and brought to you by Miltenyi Biotec. To learn more, visit www.miltenyibiotec.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box to the left and click Submit. We'll answer as many questions as we have time for at the end of the presentation. You may also use the Ask a Question box to let us know if you're having any trouble seeing or hearing the presentation. I'd like to now welcome our speaker for today's webinar, Dr. Zahra Labbaf, expert for imaging at Miltenyi Biotec. For a complete biography on our speaker, please visit the Biography tab on the top left of the screen. Dr. Labbaf, you may now begin your presentation.
Hi everyone and welcome. Today I will talk about 3D imaging of large biological samples using light sheet microscopy. My name is Zahra Labbaf and I'm a product manager for the UltraMicroscope light sheet system at Miltenyi Biotec. In this talk, you will learn about sample preparation and 3D imaging of large and complex samples such as a mouse brain. But before diving into this topic, let's take a step back and see why do we even need 3D imaging and what are its advantages compared to 2D imaging. Although 2D microscopy is a powerful tool for observing and analyzing biological samples, however, it has its limitations. One of the obvious limitations of 2D microscopy is the lack of depth information. When you look at a specimen in 2D, you can only see the top surface. This means that you might miss important structural details that are located in the deeper layers of the sample. And this is particularly important in the field of biology, as many processes and interactions occur in 3 dimensions. And without this 3D information, it can be really challenging to fully understand and analyze such processes. A major difference between 2D and 3D microscopy is the sample preparation step. For 2D microscopy, sample preparation involves sectioning of an opaque specimen. Let's take the mouse brain as an example here. So the first step would be physical sectioning, then mounting these sections on slides, followed by immunostaining of the individual sections, and finally imaging. There are certain advantages with this method. For example, you can image an opaque sample, and as you're imaging a thin tissue, the imaging might be easier and faster. And of course, there are many well-established protocols for 2D imaging and sample preparation. However, there are also major drawbacks. One is that since you're doing physical sectioning, you might introduce artifacts and compromise the sample morphology. And as I mentioned before, due to lack of depth information, you cannot really capture the complete 3D structure of the sample. And of course, since you have to do sectioning, mounting, staining of these individual sections, you would have a high manual workload. And at the end, it might also happen that during sectioning, you would lose a section, and therefore you would not be able to observe a specific structure, and in principle there is a potential to miss information. Now, what if we could do 3D imaging of a large clear sample to have a holistic view of our biological sample and skip all the hassle of tissue sectioning and the challenges that come with it? Let's look at an example. Here on the left, you see 2D imaging of a section of a mouse brain. These red structures are blood vessels labeled with an antibody against CD31. As you can see, the information that you get from this image is really limited. Now, if we do 3D imaging on the same sample, so a mouse brain that is labeled with CD31, what you would see is this. As you can see here, the difference is significant. You see a full picture, and this allows for a better understanding of complex biological structures such as brain. And of course, this is crucial to properly understand the architecture and function of such organs. Now let's have a look on the different steps that are needed for 3D imaging. A general workflow for 3D imaging involves several key steps. First step is immunostaining, followed by clearing of the whole organ, and finally image acquisition using the light sheet microscope. At Miltenyi Biotec, we offer a solution and support you through the complete workflow. We have validated antibodies for 3D immunostaining that are specific for neuroscience and immuno-oncology applications. We have the max clearing kit that is complemented with sample-specific clearing protocols for clearing of biological samples. At last, for 3D image acquisition, we have 3 light sheet microscopes. However, due to time restrictions in this talk, I will only focus on the UltraMicroscope Blaze. In the next slides, I will go into more details of each step of the 3D imaging workflow. Let's start with the first step, that is immunostaining. So how is sample preparation for 3D imaging and what are its challenges? If you remember, for 2D imaging, the sample is sectioned, and immunostaining is performed on these thin sections. So 2D immunostaining is quite straightforward, as the antibody can easily penetrate thin section and bind to its target antigen. However, for 3D imaging, the story is quite different. Here we have to label the whole organ. Therefore, 3D immunostaining of large samples, such as a mouse brain, in comparison to a thin slice or a thin section, is challenging, and that is due to antibody penetration and the difficulty of achieving a homogeneous staining throughout the entire sample. In addition to this, for imaging deep within the tissue and to acquire 3D data, we need to reduce the sample opacity by tissue clearing. Tissue clearing itself can also be quite complex, and that is due to long optimization of protocols and high number of different protocols that exist for different tissues and for different applications. So overall, the sample preparation step can be quite complex, and it might take weeks to months to establish a staining and clearing pipeline within your lab. However, don't worry. At Miltenyi Biotec, we provide validated 3D immunofluorescent antibodies together with clearing reagents and protocols to make it all simpler and save your time. Now let's have a look together at one of Miltenyi 3D immunofluorescent antibodies in action. What you are looking at is a Parkinson's disease mouse brain labeled with an antibody against tyrosine hydroxylase. This antibody labels dopaminergic neurons. As you can see, the staining is quite fascinating, and one can even see and visualize neurons that are deep within the tissue. The second step of the 3D imaging workflow is clearing. Now let's see how tissue clearing works and why do we even need to do tissue clearing. For 3D imaging, light has to pass through the sample. However, tissues are inherently opaque and light cannot pass through. The reason is that tissues are consisted of molecules with different refractive indices, such as water, proteins, or lipids. Therefore, as light travels through the tissue and it encounters these different molecules with different refractive indices, it gets scattered, and this leads to lack of transparency. Therefore, the aim of tissue clearing is to match these different refractive indices within a tissue. This can be done, for example, by removing, replacing, or modifying the tissue composition using chemical reagents while preserving the structure of tissue. To understand better how tissue clearing works, let's have a look at the following video. What you will see in this video is a glass ball that contains polymer beads. At the beginning of the video, these polymer beads are surrounded by air. However, when water is added, these polymer beads become invisible. The reason is that water and the polymer beads have a similar refractive index, and therefore light can pass through and is not scattered. Tissue clearing also works in the same way and unifies refractive indices within a tissue, and therefore light can pass through and tissues become transparent. As I mentioned before, for tissue clearing at Miltenyi Biotec, we have the max clearing kit. Here you can see a nice example of how an adult mouse brain hemisphere becomes clear over 6 hours using this kit. After sample preparation, now it's time for 3D imaging using the light sheet microscope. As I mentioned before, in this talk I will only focus on the UltraMicroscope Blaze and introduce to you some of its amazing features. The UltraMicroscope Blaze is a fully automated light sheet microscope that can be used for imaging of biological samples at a subcellular resolution. In this microscope, samples are illuminated from the side using focused light sheets, and the emitted fluorescent light is detected by a camera that is located perpendicular to the illumination path. So the illumination and detection paths are separated. Now, let's have a look at the principle of light sheet microscopy and its advantages. So, in a light sheet microscope during excitation, as you can see here, only one focal plane is illuminated at once, and therefore compared to a confocal microscopy that the sample is excited point by point, excitation in light sheet is much faster. Furthermore, due to the fact that you are only illuminating the part of the sample that you're imaging, therefore you will have less photobleaching along the z-axis. During detection as well, the light sheet is much faster than a confocal microscope, and this is due to the fact that the camera immediately captures the entire field of view at once. So overall, a light sheet microscope is faster and more gentle. So how is the full 3D sample imaged? As you can see here, in the UltraMicroscope Blaze, samples are located on a motorized stage that moves the sample through the light sheets that are illuminating the sample from the side. So as the sample is moving through the light sheets, pictures are being taken and a 3D image is being reconstructed. In the next slides, we will see examples of samples that can be imaged with the UltraMicroscope Blaze. With the UltraMicroscope Blaze, you can image samples with different sizes. You can image a sample that is very large, for example, an entire mouse model, as you can see here. Or you can image multiple samples. These samples can be imaged sequentially without the need for manual intervention or any monitoring. Imaging of different sample sizes with the UltraMicroscope Blaze is only possible due to its different sample chambers. So the standard UltraMicroscope Blaze comes with a sample chamber or a cuvette that has an imaging volume of up to 35 cubic centimeters. With this chamber, you can image multiple rodent organs or organoids. In addition to the standard cuvette, we also have an XXL chamber for the Blaze, which allows for a 51% increase of imaging volume. With this chamber, you can fit samples as large as a human kidney or even a whole adult mouse model, which I showed you on the previous slide. One of the unique features of the UltraMicroscope Blaze is that it has an automated objective changer. So you can simply go from one lens to another and increase the magnification with just one mouse click. It also has an autofocus feature and an automated sample release, which allows for easy sample exchange. Overall, these features make the microscope easy to use and reduce the hands-on time, making sure that you would have a convenient imaging process. As you learned so far, light sheet imaging is a powerful imaging technique, and it can have various applications in neuroscience research. Here we have the entire vasculature of a mouse brain. The labeling is an antibody against CD31. The unique capabilities of the UltraMicroscope Blaze and light sheet imaging make it a valuable asset in advancing our understanding of the brain and its complex functions. Therefore, this method is well suited for studying neural structures and disease models, such as neurodegenerative diseases and brain tumors, and to gain insight into their mechanisms and potential treatments. At Miltenyi Biotec, we have tools which help with light-sheet imaging in neuroscience and preparing brain samples for 3D imaging. On one hand, we have a novel antibody portfolio for neuroscience, such as antibodies against the myelin basic protein or tyrosine hydroxylase. These antibodies are validated for 3D immunofluorescence. On the other hand, we have the MAX clearing kit that ensures optimal immunostaining and clearing of tissues for 3D imaging analysis. In addition to this, we have clearing protocols specific for the mouse brain, or with preservation of endogenous fluorescent protein signals. Furthermore, we have specific protocols for clearing of organoids. Find out more about our clearing protocols, please follow the QR code. At last, I would like to finish my talk with an example of a sample that you would get following the 3D imaging workflow of Miltenyi Biotec. Here we have a brain organoid stained with different 3D immunofluorescent antibodies labeling different structures, such as neurons, neural progenitor cells, and proliferating cells. As you can see, with the UltraMicroscope, you can capture high-resolution 3D data. Therefore, it can be used as a tool for advancing our understanding of brain organoid biology and its development. I hope that I could provide you with an overview on light-sheet imaging and its applications in neuroscience. Please follow this QR code to get to a free copy of our new guide to tissue clearing for 3D light-sheet imaging. Thank you for your attention.
Thank you again, Dr. Labbaf, for your time today and your important research. We would also like to thank Labroots and our sponsor, Miltenyi Biotec, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by our speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand. Labroots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, take care, everyone. Goodbye.